Hi, Evan. Thank you so much for talking to me. In your role as a Chief Medical Officer at Applied Therapeutics, you carry a lot of responsibility, I believe. And now there's a new trial coming up for CMT SORT, which is so exciting for all of us, the whole CMT community. Yeah. How do you handle that pressure?
I think for me, I've always just focused on the patients and what the goal is at hand. So for patients who have no treatment option, getting anything across the line is huge. And especially for CMT, if we're able to get an approval for our drug, that actually will help all CMT subtypes. People have more interest in investing and doing more research and doing studies. So I just think about the patients in the end goal, but yeah, it's tiring sometimes.
Yeah, I believe that. So you mentioned CMT. The problem is right now that at this moment, we're talking in October 2025, there's no approved pathomechanism-based treatment. There is treatment, but nothing that is really targeting the mechanism of disease. So you have seen other drugs in the development, also for rare diseases. What do you think is special about CMT, and what are the main obstacles a drug has to take in order to make it to the market?
Yeah. So first is just disease understanding. So people have to understand how it is, what the mechanism of action is. And for something like CMT, there's such a variety of different pathophysiologies underneath that. So with CMT SORT, we have a clear mechanism of action. And then you really have to have a robust way of addressing that pathophysiology. And if there's something you can change to improve it. So small molecules are great because if they work, they're not thinking about a genetic intervention or something else that may be more invasive. So if you're able to find a small molecule that targets that pathway, it's really huge. And the rare diseases I've worked in before, like cystic fibrosis, those are really amenable to that. So if you have a problem with the enzyme or a protein like CMT SORT has, where you can inhibit a protein, or cystic fibrosis, we can make a protein work better. Those are the targets. But without more research within the other subtypes of CMT, it's hard to know exactly what those pathophysiologies are to find a target.
Yeah, that's very true. And especially for CMT SORT, since the gene discovery was only 5 years ago, and that's really recent, in my opinion, we still need to define outcome parameters. We still need to understand how fast disease progresses. How do you deal those issues?
Yeah, so I think the biggest thing is to look at the community, like the physicians and the patients, and what's already been developed, if anything. Some rare diseases, like the others we work on, don't have any disease-specific measures. Others I've worked on, like cystic fibrosis, do, and CMT actually does. So with the CMT Health Index and the CMT Functional Outcome Measures, or the CMT FOM, those are actually validated in the disease. So we already have tools available that are specific for that disease, because a lot of patients, especially with a chronic disease, if you use standard tools, they accommodate. They're used to how they're feeling every day, and they don't pick up on what their true disease burden is. So it's really critical to have those disease-specific measures. Now, if it's already been developed, that's great. But if you're looking to do research or have a potential treatment, you have to help develop those disease-specific measures, or find the right one and make sure the agencies, whether it's the EMA or the FDA, agree with that approach. So there's a lot of work to get there. But if you are able to have those disease-specific measures like CMT has, you're already kind of ahead of the game.
Yeah, that sounds very reasonable.
Yeah.
And what about sorbitol levels? This is a specific biomarker for the disease. How do you rate its role, like now, in the process of serving as a biomarker?
Yeah, so I think one of the keys for a biomarker is it easily measurable, and is it easily reproducible, steady over time? And then is your intervention going to change it? And is it so different from what the normal is that you're able to catch something? So with CMT SORT, the sorbitol levels are so grossly elevated that just a urine test, you're going to see it come out and be high. So if you have the symptoms of peripheral neuropathy plus that, you kind of are all the way there. And the genetics are very complex for CMT SORT in particular, hence it only being recognized genetically 5 years ago. From that, you have to have a reliable measure. We've used whole blood sorbitol. A lot of the literature and other work that people have done uses plasma, but whole blood's a little more steady state because it accounts for those red blood cells. So you have a reliable marker that you can measure over time. You know how it performs, which we do in patients who haven't been treated with drug. And it wiggles a little bit, but overall it's steady over time. So if you're able to see that change, look for that change and see if some other effect happens, then that's really what makes a biomarker good and reliable for a disease, whether it's diagnostic or helpful to determine whether you're having an effect.
Yeah, great. And now we're here all together at a conference in Antwerp. So we are all united, patients, also clinicians, basic scientists, and of course industry partners. So how do you rate the role of a conference like this? Is it important? How can this drive the process forward?
No, these are really critical. I think for me, I think of 3 pillars, and I was trying to find an outcome, a treatment for a disease. There's the patients and what they're experiencing, what they need to have done. There's the academics and the physicians and the treating people who know the disease, who understand the disease, and then there are industry partners who are able to help get those projects, those studies done. And the 3 of us together is what you need. That's why conferences like this, that brings us together, that shares knowledge, that lets us express our own voices and come together to work together, because we can't work in a silo. We all have to be in the same community, and it takes all of us to make progress for patients.
Yeah, I couldn't agree more. Yeah. Thank you so much for making the time, and have a nice day in Antwerp here.
Thank you, Maike.
Bye.
Bye.